I'm Monica, and I wanted to just share a couple of announcements from the steering committee before we introduce and welcome our speaker for the day. So without further ado, the first announcement that we had from the steering committee is regarding the regular webinar schedule that we have every month. We've decided to move to a new selection procedure where the working groups, each working group will be responsible for deciding and scheduling one presentation and one discussion webinar per year. Earlier today I sent out a link to a sign-up sheet for the working group leads. So for everyone here, you may hear from the lead of the working group that you're part of in the near future to discuss what webinars and discussions might be useful for you. And the idea behind this was to balance the kinds of webinars that we're showing for AgBioData to make sure that all of the different interest groups and all the different working groups are well represented. Here's on the right, I have a list of what's currently scheduled coming up in the next couple of months that Jacqueline and Laurel and I have been working on scheduling. And there's a couple of holes that come up in September. So just so you know that that's going to come up. And if you have specific suggestions for your working group, go ahead and get in touch with your working group lead. Next up, so we wanted to talk a little bit about funding updates. As most of you probably know or remember, we applied for a NIFA CIN grant, and we haven't heard anything yet. We recently heard that the panel that was expected to meet, I believe in February, won't meet until June. There's been disruptions that we all are quite aware of. So the steering committee is deciding what to do, and we're going to meet after this meeting and tomorrow to discuss next steps, including the possibility of resuming work on the NSF RCN Research Coordination Network proposal. So we'll try to keep you up to date on that as we hear more. And steering committee, make sure you stay on after this. Finally, just before we go on with our speaker, we seemed kind of remiss to not address the elephant in the room that we're dealing with a major new situation with the coronavirus, and we just wanted to reach out to the groups to make sure everyone is doing okay, and maybe ask how people are coping with the new situation, and maybe dealing with new opportunities perhaps for databases. One of us brought up that the International Society for Biocuration has been discussing what opportunities or projects that we databases could offer to bench scientists who are not able to go to the lab during this crisis. Are there any other sort of projects that people could help set up to capitalize on all this talent that can't be at the lab right now? And so one of the suggestions here, at least the I5K workspace, we're working with people to help transcribe our webinars, but also considering advertising and offering more manual curation through our Apollo service for people who have the insect genomics expertise but can't go to the lab right now. So we have maybe, I thought I might open the discussion for 5 more minutes before we start with our speaker. If people have any ideas about that or want to bring up anything else before we go on with the main event of today. Does anybody want to speak to that?
Well, I can mention, this is Marcela, hi.
Hi.
We had, so the Jamboree, annotation Jamboree that we had scheduled for prior to the May meeting, we held it over a 2-week period virtually. It was 4 sessions of 3 hours each. And it was like an online curation using Apollo. And we're continuing to meet monthly for a webinar. We are also trying to recruit people because, as it was discussed in the curation mailing list, a lot of scientists are unable, not being able to go to their labs, to a bench. They want to feel, contribute. And so I'm giving an in-house webinar at our plant department next week. No, in a couple weeks.
Mm-hmm.
And I will be also offering this as a tool for people to try to curate their genes of interest during this time. Okay. It's going to be like a recruitment type of webinar.
Yeah. Okay, great, Marcella. So if any of you know scientists who are interested in maize annotation, they can go talk to Marcella. Anyone else?
So it sounds like a really good idea, Monica. This is Kevin. And what Marcella said, is there a way that you're going to keep track of people contributing, or what? Are you suggesting that?
We in both Apollo and the other tool that we have to curate genes, people put their email addresses. So we do keep track of every contribution.
Yeah, it's kind of built into the system.
Mm-hmm.
Yeah. Okay. I thought maybe that you were looking for also people willing to sign up for a project they might not normally be associated with and volunteering to get involved in another project, particularly as part of training or something else.
Right.
Anyway, yeah.
So we actually have an initiative. So the Jamboree was directed for PUIs and other faculty or students that are interested in teaching, because we try to design cures, the course-based research experiences for classrooms, undergraduate classrooms. So we had about 5 out of the, like 25% of the participants were faculty. And the aim was to design projects, student projects. So either, we have people interested in specific like pathways, or I don't know, groups say nitrogen use efficiency or some other topic in plants. And so they come up with like gene lists from like the literature, or using one of our resources, like, you go to Plant Reactome and get a list of genes in a pathway, or some other, or CGIAR, like some gene tree, like in Arabidopsis, or to Gramine, get the Arabidopsis, get the maize homologs of the Arabidopsis genes that you're interested in, and so on. So they have the freedom of choosing what genes to curate, but they can design their own their own lectures, their own paces. And we have meetings also with PUIs like every other week to see how the progress is going, how and whether they need help from us, like to curate or provide some other materials. We have gone to even they invited us to like universities to talk with the students directly and stuff in the past. Now I guess we can still help them virtually. So that's the idea.
Okay, great. Thank you, Marcella.
Tanya,
yeah, that's a great idea. Maybe we can chat offline if people are interested in capitalizing on what you've already done. I see a chat from Tanya. She says that TARE ran a webinar last week on using phylogenes for gene function inference, and made the recording available on YouTube afterwards. So yeah, thanks, Tanya. It's another great idea. Maybe we can, if people want to collate ideas or have similar experiences, maybe we can chat about that later. All right, thanks everyone. I don't want to take up any more time of Medha's. So with that, today's presenter today is Dr. Medha Devare, and she's going to talk to us about the CGIAR GARDIAN platform, which stands for Global Agricultural Research Data Innovation and Acceleration Network. So a bit of a mouthful there. Medha is a GARDIAN architect. She also helps lead efforts at CGIAR and their 15 data centers to better manage and organize their knowledge to make it fair, discoverable, accessible, interoperable, and reusable. Medha has a master's in microbiology from Cornell, as well as a PhD from Cornell in crop and soil sciences. And we invited Medha to talk because we think that there are many things that our group and our various databases can learn from her about Guardian. Some examples come to mind are the Guardian's FAIR compliance metrics and text mining approaches to enrich metadata. So with that, I will stop sharing my screen and hand the baton over to Medha. Thank you. And also, if people are not muted, if they can mute. Thank you.
Hi, everyone. I will turn off my camera, but I just wanted to say hello and give you a good look at what I look like in these days of COVID, where, you know, I'm not paying much attention to what I look like.
Good point. Thank you.
Many of us aren't. All right. So with that, I'm going to turn off my camera and get going on the presentation. I hope you can see my screen right now.
Yes, right now it's, there we go. Excellent.
Good.
Thank you.
Excellent. So Monica did a great job on telling you what Guardian is, so I don't have to go through the entire name again, which is great. I lead the organized module of the big data platform. That's we have three modules. Organize is about organizing data, as the name might indicate. And we are focusing on FAIR, but we're trying to adopt a very much a sort of a problem-based approach, a demand-based approach, as I think all of us probably are trying to do. Some of the questions that we probably get, these might seem very familiar to you, although you might be working more on the genetic and genomic end of things. But for us, we work all the way from the genetic to the landscape scale and everything in between at CGIAR. So some of the kinds of questions we face: where can I find data on nutritional status of women? What does variation in crop response to fertilizer look like for sub-Saharan Africa? I need data for my crop model. Where can I find that? I've been told to make my data fair. I have no idea how to do this. Can you help? I want to assess land use change as a climate change adaptation strategy at both national and subnational level, somewhere, maybe South Asia, maybe West Africa, wherever it is. My organization has no repository, but I would like to upload my data sets and see them get reused and derive some extra value from them. These are the kinds of questions that we as researchers, I think, either ask or are often asked. And so what we're trying to do is address all of those in different ways, but through the Guardian data ecosystem. It's not really just a discovery portal anymore. It's become much larger than that. So we focus on at CGIAR and through the organized module on encouraging the generation of not just open assets, data assets, but findable, accessible, interoperable, reusable, fair data assets. And you're very well aware of what that means. So I'm not going to delve much into that. But this is something that we're very much working towards, with the proviso that increasingly we're also having to confront ethical data. So in fact, this should probably be FAIRE for the ethics dimension, with the increasing moves towards taking care of personally identifiable information and all of this. And this is particularly important to us in socioeconomic and agronomic data and a number of other data types as well. Any organization that's surveying people needs to be taking care of these concerns. So that's something that we're also focusing on. I won't talk too much about that today, though. Right. So the Guardian data ecosystem, what does that look like? Well, at the heart of it is the sort of the data discovery portal. And when I say data here, I mean data assets, I mean publications and data. And so what we're trying to do is not harvest data sets, but just point to data where it sits. So making data discoverable wherever it sits. And right now we're looking at a multiplicity of several different data repositories, at least 30 across the CGIAR centers, 15 data repositories and 15 publication repositories, as well as a number of other repositories that I'll mention as we go on. And what happens is that we harvest the metadata. So we are basically creating a large metadata index and trying to present all of that information in consistent ways with added functionalities, which I will also show you as I move through the presentation. So that's kind of the heart of this. What we're hoping to do as we get more and more semantically enabled data through the use of ontologies, as we keep going. Right now, many of our centers are using controlled vocabularies, but not at the data variable level. We're trying to encourage more of that. And so once we have a rich data pool of semantically enabled data, we will be able to query that data through semantic means, and that's very powerful. We're already producing some neat data products, and we intend to keep that going, which will be available for search engines as well as external apps, which are more sort of facing the farmers and the policymakers, much more so than Guardian itself or any part of this ecosystem probably. So that's kind of the heart of the whole data ecosystem. Then there are these two cogs at the very top. And there, what we're trying to do is enable the generation of semantically enabled, very well described, interoperable, ideally interpretable data assets, really. So I'll show you a little bit on both those pieces. And what we can do once we have well-described data, interoperable data, is to be able to explore that data. So we're also building sort of increasingly data exploration pipelines. We aren't at the stage right now where we can do this dynamically. Well, we can do some of it dynamically, but we would like to be able to, through a couple of different clicks, get to the kind of functionality you see through NCBI, for instance. That's really, for me, the holy grail. And for those of us involved in this work, we really want to get towards that, but we're dealing with extremely heterogeneous data at very many different scales, and so it's a very big kind of can of worms that we've opened, really. Right, and then lastly, what I'll end the presentation with is very hot off the presses sort of functionality for analytics. We're trying to build also relatively seamless analytical and analytical pipelines, really, including some to popular crop models. So I'll show you a little bit of that. Right, so I'm going to jump in and start with what I call the heart, which is the Guardian Discovery Portal. And what that looks like is this. Here's the URL, it's guardian.bigdata.cgr.org. Currently, we have over 155,000 publications and 23,000 data sets of varying types and values. That's for you to sort of poke around a little bit. And I should say that we're always open to feedback. There is a feedback form on Guardian that you can get to when you click on the About Guardian on the right-hand side there. So if you have any feedback for us, we'd love to hear it. Either directly, you can email me directly, or use the feedback form, either way. So I'm going to do a search like this, very cross-cutting, very broad-ranging, nutrition in women. It's something that we deal with a lot at CGIAR, and the reason I like to show this search is because it allows me to showcase what Guardian is doing most effectively. So when I click on that, when I do that search and hit return, I see something like 700 and some publications and almost 250 data sets. The screen is probably too small for you to see clearly, so I've tried to point to what we're doing here. I'm now on the datasets tab, not on the publications tab. The two tabs are at the top there. And what I see is publications from IFPRI, ILRI, and those are two of our CGIAR centers. I think we're at 13 now because a couple have merged. We're also seeing datasets from DFID, or UKAID, and from SIP, which is another CGIAR center, from the USAID's Digital Data Library, and from the Indian government's open data portal. And what we've done is gone after the agricultural data sets there. Most of it is sort of broad scale, large scale survey data, but we wanted to be able to bring that in and create a sort of a knowledge base. We also have data from the World Bank's right now we're focused on the LSMS data, which is the Living LSMS. I used to know this. It's eluding me right now. The Living Standards Measurement Survey, that's what it is. And so that's kind of microdata that we also are pointing to in Guardian. We also have data sets coming in from USDA's AgData Commons. So, wherever the data sits, we're trying to make it discoverable. Again, we're not sort of harvesting the data whole scale and bringing it into Guardian in some way. That's not our business. We also at the top here have a way to filter the data that's returned to you in multiple different ways. And you can also hone in on by theme, essentially, or by the top hits by year, or by the top countries, etc. So there's different ways of slicing and dicing this data that we've tried to provide. Now, when I click on any one data set, and I go to see it, this is a data set from SIP, our International Potato Center, and all of the data sets look the same way. So that's already sort of an added value that all of these data sets or publications, no matter where they reside, we've sort of used the metadata and built these similar looking views for the user. We've tried to include machine readable licenses, or links to machine readable licenses where they exist. And the DOIs that you see there under links, you'll see either a handle or a DOI that will actually take you to the resource, so it takes you back to the repository. We have linked the data within Guardian, so that when you click on an author, you see other publications or data sets that person has authored. And then you can also hone in on particular geographies. In the left-hand panel there, you see a bit of a summary or an abstract of that exists. We're not creating that, but we are using it to enrich the metadata that we already get from the repositories. So anything that comes with, that we can lay our hands on, essentially through Guardian, is being used to enrich the metadata. That's behind this. Now, if I go further down in that view, you see the sort of the FAIR metrics associated with each resource. This is not in any way to try and monitor what people are doing. It's just a way to sort of give an indication of how fair this is and what you might be able to do with it, and give the data provider also some indication of where they might want to focus their energies if they want to improve their FAIR metrics by Guardian standards. Now, Guardian uses the Dutch National Archives DANS scoring method, but has its own little twists on it. And it's not perfect by any means, the FAIR metrics, but it does provide one way of looking across the entire sort of data pool that's in Guardian and getting some sort of broad understanding of how fair things are. You see in the middle this keyword cloud. That is, to the extent possible, coming from it's ontologies and controlled vocabularies that are being used primarily at CGIAR, really, to annotate in the metadata fields, basically to tag the data sets or the publications. And then when the data set is completely, it's unrestricted, so it's CC0 or CC BY, you have a direct link to the data set files, but only in those instances where it's CC0 or CC BY. Then at the very bottom, you see relevant publications when you're at a data set page. And that is being built in Guardian. So we have algorithms that are, the bigger the data pool and the richer the metadata, the stronger the algorithms. It's not, again, 100%, but it works pretty well right now, so that it gives you some context, which is really important. Oftentimes, data repositories are very distinct from publications repositories, and there's not always a link between a particular data set and the publication that uses that data even. Increasingly, that's happening at CGIAR, but it's not always happening elsewhere. This allows you to sort of get a sort of a bigger context, bigger picture view on a particular resource that you're looking at. So that's what a Guardian page looks like. This is for data. The publications page is somewhat similar. I talked about standardized data collection tools, and we focused primarily on the agronomic agronomy, really agronomy experiments, agronomy trials, because the breeding folks at CGIAR already have tackled this. They had a head start, and they have field books that are based on semantic standards, ontologies primarily, really, to realize this. So we went ahead and decided that what we needed was an agronomy field book type thing, and partially this came out of my own work before I came to, well, actually, while I was at CGIAR, I started out at CIMMYT in Nepal as an agronomist. I am an agronomist by training. And it was extremely frustrating trying to get even just data from my project, which was just focused to one country, 8 districts in one country, and I still got a really kind of messy data, no matter how hard I tried to standardize the data templates. So I was thinking, well, perhaps we can just kind of, you know, get people to collect data digitally using already standardized templates, you know, where people can't muck around too much with the column headings and the data variable descriptions. So this is what we focused on. And that I just gave us a webinar on that yesterday, so I'm not gonna, you know, that's a whole webinar on itself probably. But what this is based on an ontology that we created with this use case in mind. So that's the Agronomy Ontology, AGRO. And the people who've been most instrumental in creating that have been Marie-Angélique Laporte and Céline Aubert at Bioversity, at the Alliance, see at Bioversity now, in a team headed by Elizabeth Arnaud, who I believe some of you at least work with. So this has been really, really helpful. It enables us to generate standardized fieldbooks, as I said, to collect agronomic data. So that means that you're standardizing data at the collection point with ontology terms, with URIs that are specific to certain data variables. So you're not mucking around with this at the very end after you've published your paper. Now, oh my God, I have to upload this data set. Damn it, I have to, you know, now expend the time and the energy in making this stuff conform to whatever the mucky mucks are telling me to do. None of that. So it's standardizing it at collection point. We've linked with the diversity arrays, a small company in Australia that the breeding platform is also working with to develop field books. And so we're working with them. We now have, we've enabled digital collection of the data by, you know, you create your field book, you export it to this KD Smart application. And then once you collect your data, you're sending it back to AgroFIMS to allow some data validation and statistical analysis, which is value add for researchers. And then since the data that's collected not only is standardized in terms of semantics, and make, you know, is interoperable, it also aligns a priori with CGIAR CGCore metadata schema, which is essentially based on the Dublin Core standard. So what we're doing here is giving researchers a lot of value and a lot of hopefully saved time, even though it takes some time to develop the field book. They would have to develop field books and protocols anyway. So what it's doing is giving them the standardization and the metadata alignment and saving them time at the flip side, on the flip side of this. I've included a link there at the bottom of this little poster to the field book. So if you want to poke around. If you want to know more, I'm here. Céline and I will be very happy to field questions on this. Céline is the project manager. She helps me manage the project. She's wonderful. So feel free to contact us with any questions you have on that. The other semantic tools. Now, what if I'm not an agronomist? I already have a data set, or you know, I am an agronomist, but I have a data set, and perhaps I have a bunch of historical data that I do want to make fair. How do I do that? So that's focusing on this little cog in the GARDIAN data ecosystem. What we're doing is building a whole bunch of fair workflows. They should be ready any day now. And what that allow people to do, it's a sort of step-through workflow that allow you to annotate your data with, so tag metadata. We're using COPO, the COPO tool for that. Many of you may be familiar with that. If you work with genetics and genomics data, that's what the COPO folks at Earlham Institute in the UK the COPO is optimized for that. And so they have now worked with us to customize for the CGIAR metadata schema, and have worked with us also on the ontology annotation tool that's built into COPO. So increasingly, we want to give that, or we're testing that, and we want to build that into our FAIR data workflows. So once you've annotated the data, you would choose a license as part of this workflow, machine readable license ideally. And then you would have access to what we call the Pii Checker, or the personally identifiable information checker, that allows you to run, it runs over the data set that you want to upload, and flags gives you an alert essentially if there is personally identifiable information. Right now that's restricted to the names of people. We also want to try and figure out a way of looking at whether the data sets contain coordinates to sort of hone in on a particular household, for instance. That would be a no-no. So we're working on tweaking that, but the Pii checker will be part of this workflow as well. And then you would be asked to basically say, yes, I certify the data is free of personally identifiable information, and I certainly don't hold GARDIAN responsible, or CGIAR, well, GARDIAN responsible really, or the big data platform responsible, if there is. And then you can do one of many things. You can either upload the data from there to a repository, a CGIAR repository or other repository, ideally of your choice. You can take the data with you, or you can say, I want to upload it to GARDIAN. I don't have any other repository. I would like to upload my now verified data to a repository that is sort of an exclusive GARDIAN repository that GARDIAN will also look over and harvest from. So that's kind of the workflow, and it will give you ideally a way to comply with FAIR metrics to the best of your abilities, and to the best abilities that the available ontologies allow, really. So that's kind of, I spent a little more time on this than I usually do, because I figured, Monica had mentioned interest in FAIR metrics, so there you go, and I'm happy to take any more questions about it. As part of the FAIR metrics, we've also made some guidance available that shows it kind of is a blow-by-blow account of how you can get from kind of a 0 to a 5 for each of these F, A, I, and R. So that's available on the GARDIAN page as well. If you go to GARDIAN Analytics and click on any of the metrics links there, the little orange buttons that say FAIR metrics, you will find your way to this eventually. And so this is what I'm showing you is just for findability, but the same kind of thing exists for accessibility, interoperability, and reusability. Right. So now diving into the part of GARDIAN that relates to sort of exploring a data set. I'm showing this data set from IFPRI, and this is work that it's basically data from a model. It's model estimates for crop production, working on crop production statistics, really from globally, from everywhere in the world. I know Jawu is on this meeting, Jawu Ku. He was responsible for heading up this work with contribution from just about all of our GIS folks at centers. So I can click on this Explore Data link here, and I get taken to, I had a video here, but I took that out. But what I can do here is when I come to Explore Data, I see sort of the list of all of our mandate crops. There are about 35 crops here. You can click on any one crop, like I've clicked on rice here in the left-hand panel, and I can choose any one of these or more of these parameters here, from yield to production, essentially, harvested area, production, and it's both for rain-fed and irrigated. I've chosen harvested area there, and then I can go to my panel here on the right-hand side, I choose my little pin here, this clicker, and it says, okay, now you have to drop the pin on any area that you're interested in. So I've dropped it on India for rice. And what I see is a box that pops up that tells me the harvested area for India, for rice. And in fact, I've chosen here rice and wheat because I went through this panel on the left with all the crops. I chose rice harvested area, and I did the same for wheat. So now what you're seeing here is a legend that shows you the rice in blue and the wheat in sort of yellow ochre. And once I drop that pin on any country, it pops up the summary statistics for that parameter that I've chosen. I can also say, actually, I'm interested in honing in even further. In this case, maybe I'm interested in in an area I know to be pretty high yielding, and where the harvested area is likely to be high of rice and wheat, which is the Indo-Gangetic Plain. So I've chosen part of the Indo-Gangetic Plain, and the way I can do that is by going to this polygon here. I hope you can see my cursor on the right, above the pin icon on the right-hand side. And when I click on my map, it allows me to very quickly draw a polygon, and I've done that. It highlights the polygon, and as soon as the polygon is completed, another box pops up telling me, this is the box on the right-hand side here, with summary statistics for that particular polygon. So it's pretty cool. I mean, this is pretty interactive. It allows you to essentially spatially query the data that's making this up. And we want to do much more of that and hone in, improve these capabilities as we go forward. Again, I'm sorry I couldn't show that to you in action right now, but you can play with it yourself, hopefully, and see how it works. We've done the same thing for a very large, this is a 7 terabyte data set, the CMIP5 data set. We will soon have the latest data, CMIP6, which is climate data that is massive. And you can explore that data set as well by clicking on that Explore Dataset button. So this is the bioclimatic indices. I don't have, again, the video is not working here for this time, but what we have is a series of about 12 models, global models. You can choose any one of these. I've chosen one there. And it's available, this climate data sets are available through the same Explore Dataset function here on the bottom. The top icon here is for the global production data. This middle icon here shows you sort of all of our gene bank accessions. It maps them for you. You can choose a crop, and it'll throw all of the accessions on the map, the global map. And this last icon here is the climate data. So you can choose something like maximum temperature or precipitation. And what we have is data for essentially the 4 commonly accepted carbon dioxide scenarios. This one's showing you for 2.6, but that's kind of a no, it's not even valid anymore. We're now looking at 8, RCP 8. So when you click on one of these, you click on one of these and you can choose among the 4 scenarios. You click on that and you can sort of interactively play the screen essentially, which will show you for the parameter chosen, in this case, maximum temperature, all, what the global outlook is for maximum temperature from about January 2030 through about 2080 or so. And the same for any one of these parameters chosen based on that particular model. You can also do the same thing I did for the global production. You can click on any one country and get sort of the max, the value for that particular country. And you can also play use the slider at the bottom here, this little slider, to go to a particular date that may be of interest and get your information of interest that way. So this is some of the sort of the data exploration, visualization capabilities that we've enabled with GARDIAN. We've also tried to build data products, and this is a very roughed out early data. The team at UC Davis that we've been collaborating with has worked much more on this and is close to publishing this work now. They used GARDIAN to answer that question about wanting to understand variation in crop response to fertilizer across sub-Saharan Africa. They found 200 plus data sets, of which I think they ended up choosing something like 130 data sets. They had to spend a lot of time still wrangling with the data to clean it, to really understand what the variables meant and whether the particular data set they were looking at had the variables they wanted. And then they were able to sort of derive some insight from that data. So the graph on the left, the very noisy one, shows you sort of yield in response to different amounts of nitrogen as derived from the data, and it is very noisy, as you can see. And then on the right-hand side, this is showing you sort of spatially sort of a map essentially of response to 50 kilograms per hectare of nitrogen. So they've taken the 50 kilogram of 50 kilograms per hectare of nitrogen and mapped the locations to the extent possible, again, very roughly. And they're seeing some interesting patterns, which, you know, they probably dove deeper to see if this was real or not, because you see something going on with Madagascar there that's sort of interesting with pretty high yields. So this is a real life example of what you can do when data is made available, when it's open. And, you know, if it were fair, if it were truly fair, all of the data, if you had a fair data pool, an interoperable data pool, you would have a much easier time and we would be able to much more quickly and agilely respond to the kinds of global challenges that come our way, really. Whether it's COVID, in this case, we don't deal with that, but you know, you could imagine a similar sort of thing for crops, for instance. Would we be able to quickly get off the blocks? Well, not really, not even now, even though we've made a lot of headway. So there's a lot of value to making the data open, to using machine readable licenses. Many of our, not many, but a couple of our centers still aren't doing that. And it's really important because, you know, without that, machines cannot operate over the data very seamlessly. So that's really important. In some cases, it's important to keep that data locked up, perhaps, but in other cases, especially if you can assure a secure environment for the data, it's really important to make it completely open and completely fair to the extent possible. And of course, with the ethics stamp on it. So with that, it leads me to collaborative GARDIAN labs. And the reason this came about is because we realized that people were having to use many different, you know, many different pieces of software to do the kinds of things that they normally did. So you might use Slack to communicate and collaborate effectively with your team. You might use email or FTP to exchange data. We're still shuttling data sets around via email or Dropbox. And you're worried about security perhaps there. You're using GitHub to manage code. And you might be using some analytics platform that's totally different for data analysis. And in some cases, it's Jupyter Notebooks. In other cases, it's something bespoke. What we've tried to do with CG Labs is to combine all of this so that it's a place where researchers can really, you know, what we're offering essentially is all of these features with seamless interlinks. At least that's the goal. We're not completely there yet, but we're getting there very fast. This is, as I said, very hot off the presses. So what I'm showing you is very new, and not all of the functionalities are sewed together yet, but they're all sort of there in pieces. Many of them are sewed together. So what we're using for the secure data exchange and transfer is developed by the University of Chicago. It's called Globus. It's a software as a service, and it is HIPAA compliant. So that means that it conforms to the strictest standards in terms of exchange of patient information even. So it's pretty secure. We've implemented that in CG Labs to be able to not only find data. So here, by entering CG Labs, and I'm in it now, and what you can see is when you log in through Globus, you see this dashboard here. You see a user manual, so that's probably a good place to go to start with, because it's quite complicated, you know, at least to begin with. But you can go here. You can communicate with very openly with folks. You can share things with people as long as it's not sensitive information. But then when I go to my personal space, I see a menu that looks like this, and you will soon see the same kind of space or the same kind of menu in a group space, because that's the kind of functionality that we want to get to, where I can set up my group, my trusted collaborators, my lab team, for instance, and I can very quickly find data. I can share data with them securely. And I can analyze that data as well. So this is and I can do some other things as well. But those are the pieces that are particularly of interest. So here's some data sets that I searched for. I did a search, and when I click on Find Data, it'll bring up a sort of a search box. And what that search is doing is essentially searching Guardian right now. But we might choose to make it broader, or rather, we might actually point to more stuff in Guardian. So that it's searching for that data. I can bring in that data to, if those data sets are downloadable, I can download them into this space, into CG Labs. And then when I want to, I'm not going to show you the security share data part of it, but I can, it's easy to use, and I can do that with trusted entities. And the analysis screen, when I go to the analysis screen, it looks something like this. So here on the left-hand panel, there's a bunch of icons here. The top one refers to the files that I've actually already downloaded, that I have available. And those downloads, I can download data from my own laptop, from somewhere else, or from Guardian, you know, from a Guardian search. So it's quite flexible that way. You're not you know, tied and locked into a Guardian search necessarily. So I've done this. I have my data sets there, and what I'm also showing here on the right-hand panel is a model run. So we've got the Wofost crop model. That's a Wageningen University developed crop model, and there's an R version of it. So we've got that in sort of, you can work with it in CG Labs very seamlessly. So I've looked at it. I've said, okay, I want to run the model with some very basic parameters. I'm looking at looks like electrical conductivity for soil. Maize is the crop, and a particular start date for data. I get a model output there, and it also throws, this is LAI. You can't see the, well, it's LAI plotted by date, by Julian date. So what you see here is leaf area index, and as you might expect, as the crop grows and peaks and then senesces, you see that kind of curve for the leaf area index over time. And that's just a very simple model run that you can do with CG Labs. So you can also take your own data set, and I thought I had a screenshot of that, but apparently I don't. You can take a data set that you've downloaded, and you can run analytics on that. You can work collaboratively with your team. That's not completely set in place yet, but it's coming very, very soon. You can share code. So there's you see the Git tab there. You can download code from GitHub. Here you can upload code back to GitHub if you made some changes. You can work collaboratively on the code as well. So it's a very nice sort of increasingly seamless environment to be able to do some of the kinds of analytics that we tend to do in agronomy, particularly. Well, it doesn't have to be just agronomy, but in the agricultural domain writ large. So that being said, I just wanted to talk a little bit. This is the entire GARDIAN data ecosystem, and I showed you sort of the organized pieces of it. But this work is very much dependent on particularly one of the other two modules, the convene module. The other modules inspire, but the convene module here hosts a number of communities of practice. And so what I'm showing you here, the two things that just, here, let me back up. The two things that just popped up are the ontology community of practice, which is very instrumental, of course, as you might expect, in the semantic enabling of what we're trying to do. So I've shown you, I've tried to put these little icons where they are most involved. So that's the ontology community of practice. We also have the community for spatial information, which is another community of practice, which has enabled a lot of the functionalities that you saw today. We have the digital data, or digital agronomy community of practice, which is also important. And we have, I can't remember which one that is. Let me just finish up. Digital data, the crop modeling. That's the crop modeling community of practice, which is, as you might expect, very much involved in the analytics end of things. And then the last one is the socioeconomics data development community of practice. And they're involved in multiple different places as well, as you see there. And then, of course, there's the data and information management community of practice, which is involved, broadly speaking, in a lot of this, but particularly in the part that allows us within CGIAR to make our data findable, accessible, interoperable, and usable. So there's a big shout out to them. And then lastly, I wanted to throw up the Inspire part of this, which is the third pillar or the third module of the big data platform, which is focused on essentially the kind of the blue sky projects that link CGIAR researchers with sort of the non-traditional, for us anyway, non-traditional folks in the digital agronomy, in enabling the digital agronomy piece of what we do. So that those are the app developers, for instance, who take what we're doing, or who can take some of the insights of what of the research that we do to the farmers, to the policymakers. So that's what the GARDIAN data ecosystem looks like. And I want to end there, and I want to say that this, while it's been largely spearheaded by the organized module, it depends very, very much on a number of different players, just a grab bag of whom are here. SIO is the team that we work with to develop GARDIAN, and we've worked with all of the CGIAR centers. We're very grateful to the CGIAR centers for the work that they do in making their data conform to a lot of these standards, and in working with us on the work that we do to organize data. And then there's particularly strong collaborations with the University of Florida on some of the modeling pipeline work that we're doing, with Rothamsted Research on agrofilms, and with the University of California, Davis, on both the analytics piece as well as the agronomy field information management system, the agrofilms fieldbook generator. So with that, I will stop here and take any questions if there's time. I don't have my watch in front of me.
Thank you, Medha. There's a bit of time. Thanks. This was amazing to see all the amazing work that your group and related groups have been doing. I thought I might kick off with a question. A lot of the databases in AgBioData, we deal with genetic, genomic, and breeding data from crops, insects, animals, that kind of thing. And metadata is often a sticking point, and a lot of our work is in wrangling metadata from our stakeholders, our users, the scientists that supply the data, and putting it in a shape for specific community standards that might differ among our databases as well, and might differ from the ones that GARDIAN is ingesting right now. So in terms of thinking about bigger picture of how GARDIAN is more of a larger network that would probably ingest data from our databases, assuming we have web services and our data are compliant enough. How can we, with the individual community metadata standards, interact with GARDIAN in the future? Is there a mechanism for that? How do you deal with individual community metadata standards if GARDIAN might not be yet tuned towards adhering to those, or maybe you are? Can you speak to that?
Well, that's a very good question, and in fact we have faced that, and we face it all the time. Every time we look at a new repository that's not a CGIAR repository, and sometimes even with a CGIAR repository, you know, we the different CGIAR center domains necessarily require some sort of bespoke, you know, development, or at least, or it might require bespoke development. But certainly when you're looking at the DDL, USAID DDL, or the World Bank microdata library, for instance, or USDA's, you know, AgData Commons, these are all different metadata schemas. And so we're harvesting all of the metadata and trying to map it to our consistent, you know, make it consistent sort of with our schema, or at least map it to the best possible to the best degree possible. So that's something that I think can easily be done, and we'd definitely be interested in working with you on that. So if you want to email me after this, I can put you in touch with the SIO team, and we can set up a meeting to, you know, to kick it off, and you can, well, actually to kick it off would be useful, and to point us to any URLs you might have, so that our developers can look, the SIO developers can look at the data and figure out what that might need. Does that sound like a good plan?
Yes, I mean, it might be, well, we'll have to talk with the steering committee individual members first. I'm kind of wondering, it might be a bit premature, and we'll just have to kind of see the lay of the land first, because we're still, one of the working groups that we have for AgBioData is to develop the metadata standards for each group. But yeah, we should get in touch for sure.
Sure. One of the things we were already doing was harvesting the European nucleotide archives. And there, you know, that you're right, that schema is very, very different, as, you know, would be the NCBI, you know, genes or nucleotides metadata, for instance. But there's no reason, I don't see a reason why we couldn't include that relatively easily. I always say this, and then luckily we're working with the SIO team, whose answer is almost always yes, we can do it. So, you know, I can see a way to doing it. Let me discuss it with them a little bit further. And in the meantime, you can do all your, you know, checking as well.
Okay, great, thank you. There's time for another question or two.
I see the questions in chat here. Do you want to read them out, or do people want to ask them themselves?
Oh yes, thank you.
We ran a webinar last week using phylogenes for gene function inference, made the recording available on YouTube after blah, blah, blah.
Oh, that was from earlier, yeah. I think the next one here, so there's a question about the data sources that you ingest. So PubMed, PubAg, Agricola.
Yeah, we actually, I should have changed that diagram, because PubMed, we were pointing to PubMed. But again, we didn't bring it into GARDIAN so that it's not visible in GARDIAN the same way that the other things are. So we need to sort of address that. But absolutely, we will take a good look at Agricola and go after that as well. Good point.
Great. There's another one from Marcella. Could you please post the link to the field book?
Oh, sure. It's in the presentation.
Okay,
but
we'll be sharing the slides, I think, later, right, and the recording.
Yeah, but I'll just go grab it and stick it in there. I'm never one to miss an opportunity. I have a captive audience, so I might as well do it.
Yeah.
Right. So there you go. That's AgriFilms.
And
that's been developed on top of the highly interactive data analysis platform at SIP. So it's a very strong collaboration with SIP and with folks at Bioversity, who are the ontology folks again. So, you know, and a number of other people at centers, because, you know, the researchers have been our sounding board for, you know, to make sure that we're actually responding to need.
Yes.
And then Sushma asks, hi Sushma, long time no see. She asks, what is the copyright on data Hi. She wants to know what copyright on data and images from Guardian. Can they be used for open book projects, teaching, and other things? Huh. Yeah, I mean, yeah, you can. The only thing I would say is the actual data sets themselves and the publications have different use rights. But other than that, the data images, go for it.
So whom to ask? Medha, I'm developing an open text for OSU students, and I might actually be interested in using some of the things from this website. So whom should I contact?
That would
Because
be
I
me.
need to provide the OSU press a note that they agree to open use of the material.
Yeah, no, that would be me.
Okay,
So,
thank you.
and I think the answer will probably be, depending on what it is.
Yeah,
I
so they
have
are
to
going
sort of,
to do the CC 4.0,
yeah.
which means that other people, after we publish our book, other people can also use that material.
Sure. If
Right.
it's just sort of, I don't know what you mean by images exactly, but if it's sort of more like screenshots and stuff, I think you can do that for sure. But just write to me and let me know what it is you're looking for, and most probably the answer will be yes, I'm guessing. We can certainly talk through it. And then
Okay.
Pankaj has a question too. Sorry, Sushma, did I answer?
Yeah, thank you very much.
Sure. Hi, Pankaj. Good to see you too. Do you use any of the Plantium ontologies? Yes, you missed a talk. I don't know if you were there yesterday for the agro talk, but we do, through AgroFilm, certainly we use, the agro itself started out as essentially a data dictionary, and then we went out and mapped to other ontologies, many of which I think are Plantium ontologies. So it's trade ontology is one of yours, right? I know Laurel's on this call too. Yeah, okay, he says yes. So yes, trade ontology is one of them. The others are unit ontology, Envo, Pato, I can't think of others. I think there's a whole bunch of them that we sort of, that the concepts in agro are using, so that we're not multiplying silos, essentially. Great.
Excellent. All right, we're already a bit past the hour, so maybe we should say thank you to Medha. This was an excellent talk, and I certainly learned a lot. And let's keep in touch. I'm sure if people have questions, they can be able to contact you, right? Absolutely. I welcome questions. I may not, I may need a kick in the I was going to say something bad. A kick in the pants. If I missed something, don't hesitate to poke me again. But absolutely, I'm yeah. Great. And the last thing I'll say is I think the talk is being recorded, but I will share the presentation as well. I've added notes to most of these slides, but the hot off the presses one, the CG Labs one, I'll just add some notes to those and share them by the weekend. Okay, thank you, Medha. Sure. Oh, looks like Marcella has one very quick announcement, right, Marcella?
Yes, just to be on the lookout for one email that about our regular bioinformatics workshop during the ASPB plant biology meeting. We hope to get it out today. So we're gonna have 2 hours prior to the start of the meeting. As of now, we don't know yet how the meeting is gonna be held. The likelihood that it's gonna be virtual or partially virtual, in person, a combination of in person and virtual. But we're just we're moving ahead in the assumption that we can do it virtually and be prepared if we need to switch. So from the results of the survey that we held to all of the participants at the last year's workshop, there was a preference to have a combination of short and long talks, but it also will depend on how many people are interested in giving a talk. If there is too many that we cannot accommodate, we are going to have to set some parameters to select, like last year, and ask people to submit their abstract and make a decision from there. But this year, it's Shabari from Phoenix and myself, Marcela from Cold Spring Harbor, are in charge of the meeting. So just expect an email, probably sent through Jackie. But that's all I want to say. Thank you.
All right, thank you, Marcela. Okay, thanks to everyone for coming. And I think the steering committee is going to stay